Good afternoon, everyone. Welcome to our Thursday afternoon webinar. I'm Melinda Bikini, the Director of Advocacy with the Cholangiocarcinoma Foundation, and we are pleased to be joined today by Dr. Thomas Schuetz, MD, PhD, who is a board-certified medical oncologist. He has over 20 years experience in oncology and biopharmaceutical drug development. Thomas holds a BS in chemistry, an MD from Harvard Medical School, and a PhD in genetics from Harvard University. He completed his medical training at Massachusetts General Hospital, where he served as the chief medical resident. He completed a medical oncology fellowship at the Dana-Farber Cancer Institute. He's going to give us a presentation on their new investigational drug. I ask you guys to put your questions in the Q&A box or the chat box, and we will answer them at the end. And one thing I ask of you is if you can kind of keep the questions general and not ask for specific medical advice, that would help us out completely because we're not able to answer those questions. And so Tom, welcome. We appreciate you being here.
Gosh, thanks so much, Melinda, and thanks to the Cholangiocarcinoma for inviting me to this. I'm super happy to be here today, and it's a real honor to talk to all of you today. And I look forward to answering some of the questions that you all may have about the presentation. So just one thing that Melinda just said is actually in my first slide. So I'm the CEO of Compass Therapeutics, located in Boston, Massachusetts, with the skyline of Boston in my virtual background here. But Compass is a public company, and I'm required to give disclaimers when I give presentations that I'll be making something called forward-looking statements, which is I'm going to be talking about things in the future that may or may not occur. And Melinda just said, you know, I'm not here to give medical advice to you all. I'm here to talk about a drug that we call CTX-009, which is an investigational drug, and I'll talk more about that in a minute. And this drug, of course, is not yet approved by the FDA. So I'm going to talk a little bit in some general terms here for a couple of minutes about some drugs that are used to treat patients with cancer. Many of you are very familiar with all of these drugs. I'll talk a little bit about the process with the FDA, just to put into some context exactly where we are today with the development of CTX-009. So there are many different types of therapies that are used today to treat patients with cancer. All of you, of course, know what chemotherapy is. And for the most part, chemotherapy are, in general, chemicals. That is, chemicals that are synthesized, and those chemicals mostly work by killing cells that are cancerous cells in your body. So over the past maybe 10 years, a new form of therapy has been invented called immunotherapy, which are drugs that stimulate a patient's own immune system to help fight your cancer. So another new category of drug within probably the last 10 to 20 years are drugs called biologics therapy. And biologics are different because those are drugs that are synthesized by living cells rather than synthesized in a test tube. So, for example, antibodies, which many of you have heard of, if you have received the COVID vaccine induces antibodies to be formed inside your body that will attack the COVID virus if you're exposed to it. Well, what we've been able to do is make antibodies and use those antibodies as drugs. And one of the most famous, if you will, monoclonal antibodies is called Herceptin, which is sometimes used to treat patients with cholangiocarcinoma. Finally, targeted therapies are also a rather new form of therapy. Those are drugs that specifically bind to a mutated protein. And some patients' tumors have proteins that are mutated. And in cholangiocarcinoma, recently, there have been a couple of drugs approved that target something called FGFR2. So one of those drugs is called Pemazyre, the other drug is called Truseltic. So each of these types of therapies that I've listed on this slide can be used to treat patients with cholangiocarcinoma. So how are drugs developed? And I'm sure many of you are familiar with these concepts, but I thought that I would just cover this very briefly again to help put into some context where we are at Compass with the development of CTX-009. So before new drugs go into patients with cancer, that is human patients with cancer, a whole bunch of tests are done. Scientists in the lab do in vitro tests to see how the drug might work. And then these drugs are generally tested in animals. That could be mice or dogs or monkeys. And the goal of those studies are to show whether or not these drugs are safe in animals and whether or not they do what the scientists think that they do. When all of that is done, you take all of that information and submit it to the FDA for the purpose of FDA allowing that drug to go into people. And when the drug goes into patients with cancer, that, as I'm sure you know, is called a clinical trial. And there are many different so-called phases of clinical trials. Commonly, phase 1, 2, and 3. Phase 1 is the first time that the drug is tested in people, and that occurs after the FDA approves that that drug can go into patients. So in general, the goal of phase 1 is to show that the drug is safe. Phase 2 is generally a study that's designed to ask the question whether or not the drug might work. Like, can we show evidence that the drug does something to the tumor? Now, ultimately, that kind of clinical trial generally cannot prove that the drug works, but you get hints that the drug might work. And then phase 3 studies are when you actually prove that the drug is both safe and effective, generally by comparing it with some other known therapy that has been given to the patients. If that study is positive, then you can go back to the FDA and ask the FDA to approve the drug. I'm going to come back to this slide in 1 minute. So there are lots of people involved in clinical trials, and I'm sure, as you know, clinical trials are incredibly complicated endeavors. And over here on the bottom right, obviously, all of you are an enormous part of the clinical trial process, as well as your physicians and many other folks, including companies like Compass, who are generally called the sponsor. And there are also people involved in the clinical trial to make sure that the data is being collected properly and that all of you, the patients, are protected correctly when you enter a clinical trial. So where are we today with CTX-009? So we're coming up toward the end of our phase 2 testing. So FDA has approved the application to bring this drug into humans. Phase 1 safety studies have been performed, and we've recently begun to test the drug in phase 2, and we've done a set of testing in patients with cholangiocarcinoma and other biliary tract cancers. And I'm going to talk now about some of the data that we've seen from our clinical trial with CTX-009. So what is CTX-009? So this drug is actually a very complicated antibody. What it is it's a bispecific antibody. And that means that it binds 2 different targets at the same time. So one of the targets it binds is called VEGF-A. VEGF stands for vascular endothelial cell growth factor. That is a molecule that inside of a tumor, it affects the way that blood vessels form inside the tumor. And VEGF-A is an extremely important target for monoclonal antibodies. And one of a very, very important drug in oncology is called Avastin, Avastin. Avastin was the first drug that targeted VEGF-A and has been shown to be effective in a large number of different tumor types. DLL4 stands for delta-like ligand 4. DLL4 is related to VEGF, but it's different. And DLL4 does some slightly different things to the blood flow in tumors, we believe. And in animal studies, the combination of targeting both VEGF-A and DLL4 has been shown to be better in animals than targeting either one or the other. So up here on the right is a cartoon diagram of CTX-009 with arms that bind to VEGF and arms that bind to DLL4. So this is the drug that we're currently testing in patients. So how has this been evaluated in the clinic so far? So I'm going to now talk about some phase 1 data and give a summary of the phase 1 data. And then I'm going to talk about some data from our ongoing phase 2 study. So this drug began its life in South Korea. It was discovered by a company in South Korea called ABL Bio. So 2 phase 1 studies have been completed. So the first phase 1 study gave CTX-009 at increasing doses to a large number of patients, about 45 patients, to determine what might be the best dose to continue studying. That is this drug safe at these doses? What are the side effects that we see? Is there any hint that this drug might work in patients? So the second phase 1 study that was done is something called a phase 1b study, where CTX-009 was combined with chemotherapy. So the 2 chemotherapy drugs that it was combined with are drugs called irinotecan or paclitaxel. So paclitaxel is more commonly known as Taxol, which is a drug that I'm sure many of you have heard about. So we saw something kind of interesting in the phase 1 study. We saw some patients who had their tumors shrink. So these are called responses. So when the tumor shrinks by 30% or more. And that was a really interesting hint of evidence that CTX-009 might be working. So interestingly, and the real reason that we're here today, was we saw something in the phase 1b study that was quite unexpected. So when CTX-009 was combined with Taxol, we had 2 patients among 4 treated with cholangiocarcinoma whose tumors shrank quite a bit. So, and I think importantly, those 2 patients, now to be really fair about this, very small patient numbers, but these 2 patients, I think, taught us something very, very interesting. So both of these patients had had several prior chemotherapy treatments. I'm sure many of you are familiar with the chemotherapy combination gemcitabine and cisplatin, which is commonly used in patients with cholangiocarcinoma. And these patients had also received other therapies after the gemcitabine and cisplatin. And both of these patients were on the clinical trial for more than a year. So that's really, really interesting that these patients could be on drug for more than a year. So one of the One of the things that I think I would like to maybe like to point out is, 30%, is a number that is commonly agreed upon to define responses. And, it might not seem like a large enough number, and I surely agree with that. And our goal is to see patients' tumors disappear and to see patients not on drug for more than a year, but to have patients go much longer periods of time. That's our goal. But these were our first hints that this drug might have some unusual activity in patients with cholangiocarcinoma. So I just want to talk about what are some of the side effects of the drug in this study before I talk about some of the hints that it might be working, because it's always a balance. Sometimes this is called a risk-benefit analysis, right? Every drug has side effects, and I just want to talk about what the side effects are with 009. So the most common thing that we saw was high blood pressure. So in about 40% of the patients, we saw evidence that their blood pressure was slightly elevated. And in 15% of those patients, their blood pressure went above 160/100. So those patients required treatment with blood pressure medications. And that was really the most common thing that we saw in our phase 1 study. I'm going to come back to that point in 1 minute. So in the phase 1b study, again, we combined 009 with 2 different chemotherapy drugs, and we treated 17 patients. And now I'm going to talk about some clinical data. So let's talk about the format of this slide. So this is an analysis called a waterfall plot. Each of the individual bars here are individual patients that were treated in the study. So there are 17 bars on this slide representing 17 patients treated in the study. The y-axis over here begins at 0. And above the line means, unfortunately, your tumor got larger on the study, so you did not benefit from the study drug. We had a couple of patients that unfortunately did not benefit. Over on the right-hand side of the slide and below the 0% line are patients whose tumors got smaller on the study, which is in general the goal, right? There are other goals, of course. We want to help patients live longer. We want to help patients feel better. But this is one of the first ways to study the drug, to see whether or not patients' tumors got smaller. And much to our surprise, to be fair about it, over here on the far right in the orange color here are 2 patients with advanced cholangiocarcinoma. One of these patients had a 61% decline in the total size of their tumors. That's a pretty substantial decline. One of them had about a 40% decline. In the other orange color here, one patient had a 28% decline. Unfortunately, one patient had a 22% increase. So this patient did not benefit. It looks like these patients may have had some benefit. And this was quite surprising to us. And here are some CAT scans from one of the patients. And all of you, I'm sure, know what CAT scans are, and you've had all of these scans yourself. And these scans are what we use to follow patients' tumors over time. So over here, we have 3 rows of scans, one in the upper abdomen, one in the lower abdomen, and one in the lung. The red arrows show this patient's tumors. So when the patient started therapy, unfortunately, the tumor had spread outside of the patient's liver. That's called metastatic, which we see unfortunately a lot. So there was one metastatic tumor here underneath the skin in the pelvis, and unfortunately a tumor in the lung. Moving left to right is time, and you can see the tumor in the liver has gotten quite a bit smaller over time. The metastatic tumor over here on the skin actually disappeared in this patient, which is very interesting. And the tumor in the lung also got smaller. Ultimately, this patient had about a 60% decline in the total size of their tumors. So again, this is an individual patient. We can't really extrapolate too much from this patient. I'm super happy for this patient, of course, because it looks like they derive some benefit from being on the study. So based on that observation, we started a phase 2 study that was only in patients with biliary tract cancers. So on the previous slide, this waterfall included patients with many different types of cancers. So in blue, we have patients with cancer of the pancreas, patients with colon cancer in red, patients with stomach cancer in blue. In gray here, this is a patient with lung cancer. So all kinds of different cancers, patients with cancers were in this study. But in the phase 2 study that we did, we only treated patients with biliary tract cancers. Those patients received 1 or 2 prior chemotherapy regimens, and those chemotherapy regimens were no longer working for the patients. They were given 009 at a dose of 10 milligrams per kilogram. That's given intravenously every other week with the chemotherapy drug Taxol. So this is something called an adaptive phase 2 study, which is a statistical term. And the success criteria for this study were defined as if there were 3 tumor reductions in the first 21 patients treated, then we would treat some more patients. So I'm going to give an interim update from this study over the next couple of slides. So this is the waterfall plot for the current phase 2 study. So what we've discovered is that among 24 patients treated so far, this update is data as of April 14th. We have 10 patients with cholangiocarcinoma and other biliary tract cancer who have had partial responses. 10 out of 24, which is a 42% response rate. So we're incredibly excited about this data. The other thing about this waterfall that is quite unusual is that every patient's bar is actually below the line of 0. So nobody goes above the line. So this is a really unusual finding. And over here on the right, the color coding here are for different subtypes of biliary tract cancer. Cholangiocarcinoma inside the liver or outside the liver, gallbladder or ampullary cancer. And one of the things we're also excited about is that we have had at least one response in all 4 types. So cholangiocarcinoma, inside, outside the liver, gallbladder, and ampullary cancer. So as a phase 2 study, this is super encouraging. And we're really excited about this data, but it doesn't yet prove that the drug is safe and effective. And I'll come back to that point in 1 minute. So this slide is a different type of data presentation. So this is something called a swimmer plot, because each of these bars looks like lanes in a swimming pool. But as you know, these are not lanes in a swimming pool. These are individual patients. Every one of these lanes are individual patients. So what this shows going left to right is time. So this shows the amount of time that patients have been on drug and how they've been doing. So at the top, we have somebody on drug more than a year. And we have a couple of other patients here approaching being on drug for almost a year. So again, something that is just really encouraging preliminary data for us. So it's also, again, super important to be complete here. And let's talk a little bit about the side effects that we've seen so far. So one of the things that chemotherapy does, it affects the cells in your bone marrow that make cells in your blood. So white blood cells, red blood cells, and platelets. And we saw those decreases in all three of those types of blood cells, probably caused by the Taxol. That's a common side effect of Taxol. We saw hypertension again. Hypertension is one of the most common side effects seen with the class of drugs that bind to VEGF-A. So we definitely see that. Some patients require treatment of their hypertension. And when patients get Avastin, some patients require their high blood pressure to be treated. Over here on the bottom, I've listed a set of side effects that occurred in one patient out of the 24. So each of these side effects occurred in one patient. So some of these are commonly seen with drugs that target VEGF-A. We saw one patient who had a perforation of their intestine. That's a serious side effect, just to be very clear about it. That is something that is seen with drugs that target VEGF-A. We also saw one patient who had pneumonia. Unfortunately, that patient died on the study. Don't really know what the real cause of that was, but we did see that. We also saw something called proteinuria, which is a side effect in the kidney, something that is also seen with other drugs that target VEGF-A. So here's where we are today. So we believe that we have some really encouraging phase 2 data. We've got 10 partial responses out of 24 patients treated with advanced biliary tract cancers. Patients have been on study for quite some time. Another really interesting observation, I think the side effect profile is really similar to what we saw in our phase 1 studies. And I think on balance, it's manageable. And just to put this 42% number into some historical context. Now, I just want to be super clear about something. The phase 2 study is not a comparative study. So we did not compare 009 plus Taxol to anything else. Those patients just got that. So it's always a caveat when you compare one trial to another. But we do have historical treatment regimens in patients with biliary tract cancer. So the 3-drug combination regimen FOLFOX, which is 5-fluorouracil, leucovorin, and oxaliplatin. Unfortunately, that only has a 5% overall response rate in patients treated after receiving first-line chemotherapy. It has a small improvement in overall survival, so patients on average live a little bit longer. but in terms of tumor reduction, it's only about 1 in 20. First-line chemotherapy, that is the drug that you would get for the very first time you're diagnosed if you can't get surgery or radiation. The 3-drug combination of gemcitabine, cisplatin, and an immunotherapy called durvalumab was reported earlier this year to have a 26.7% overall response rate. So one of the things that unfortunately happens in oncology drug development is we saw this 42% response rate in a single arm study. And unfortunately, a lot of times when you do a randomized trial, the response rate sometimes goes down. Don't really know why that's the case. It just happens. But this number that we have here, 42% in patients treated after 1 or 2 prior chemotherapy regimens, is a super encouraging finding for us. And because of that, later this year, we plan to initiate a randomized trial where we're going to compare 009 plus Taxol against Taxol. And the design of that study is incredibly simple. 2/3 of those patients in the study are going to get 009 plus Taxol, and 1/3 of the patients are going to get Taxol. Now, this study has not yet started, so it's not. There are no open sites for this study. We hope to get this study started later this year. We're in conversations right now with the FDA about this study, and we hope to get this study started later this year. But as of right now, it's not open for enrollment, and we'll maybe update the Cholangiocarcinoma Foundation when this study is open for enrollment. So that's my last slide. I'm just going to go back to one last slide, which just has the summary and our next steps. I'll thank everyone today for your time today. Really appreciate your time today, and I'm certainly happy to take any questions that you might have.
Thank you so much. That's exciting data, but not without side effects as always. And we did have some questions come in for you. Can you talk about why you think the drugs were more effective in the 2 patients out of 4? I think that was the phase 1 trial. And were there any similarities, differences?
Right. That is a fabulous question. So the short answer to the question is I don't know the answer to that yet, but that's something we're actively studying. What we're doing is we're looking at some biopsy specimens from those patients to see if we can discover whether or not there are any subtle differences in the patient's tumors that might help us figure out which patients might do better than others. It's a super important question, and it's something that we're studying now.
Okay. And you said recruitment has not opened for the clinical trial. And so I don't know if you have an NCT number. They were looking for that. And
Sure.
do you have any ideas of where it will be opened, what countries, or can you give us any clue?
Sure. So in terms of our patients still being recruited for the study, the answer to that is no. The phase 2 study is not being recruited for. And the upcoming study, we don't have an NCT number yet for the study. So the study is, we're going to have that soon. And it should be up on clinicaltrials.gov very shortly. And Melinda, when it's up on clinicaltrials.gov, perhaps I'll just let you know that.
That sounds good.
Okay.
Okay, so the next one, if neutropenia is one of the most common side effects, are there steps in the protocol to step down the Taxol dose but stay on the study?
Yes.
Awesome.
And that's a super important question also. So, and that's something that we would definitely plan to do.
Okay, and I think you touched on this, but I'll ask anyway. For the planned study, why isn't there a CTX-009 only arm? Do we scientifically believe that paclitaxel is necessary in combo for the effect?
Ooh, boy, that is a really complicated question. So one of the things about Avastin that has been known for quite some time is by altering the blood supply to tumors, Avastin probably also alters the way that chemotherapy gets to the tumor. And so it seems like there's something unique about the combination of Avastin with chemotherapy. So we believe that there is something unique there about the combination of 009 with Taxol. Now, we have not yet proven that for sure. It's an interesting question. Ultimately, I think we may want to study 009 alone without the Taxol. In the upcoming study, we wanted to try to keep it as simple as possible. So it's a very simple, straightforward study design, to try to show that 009 is clearly adding something to the Taxol. So one of the most important steps in drug development is, when you do a randomized trial, is to show that one drug is adding something, to the other drug, especially when that's an experimental drug. And one of the things, FDA has been, fairly consistent, in their, recommendation, that companies do randomized trials earlier. And recently, FDA has also discussed in some public forums, their recommendation that randomized trials be done earlier in drug development.
Okay. Next question is, do you think this will have any impact on MSI high cholangiocancers, and can it be combined with immunotherapy treatments?
Wow. Wow, that is.
We have a good audience here.
You all are on fire today. This is a super complex question. So yes, some fraction of patients with cholangiocarcinoma are so-called MSI high. MSI, for those of you who do not know, stands for microsatellite instability. And that means that your tumor is having more trouble repairing its DNA and has more mutations in the tumor. And when we see that, immunotherapies are generally more effective in those patients. So patients with cholangiocarcinoma that have MSI high tumors will generally get an immunotherapy, so a drug like Keytruda, which is also known as pembrolizumab. And Keytruda can be very effective. Keytruda is an approved drug for patients with MSI high tumors. Now, one of the things that has been shown scientifically is that VEGF-A blocking drugs alter the migration of cells of the immune system into the tumor. So in patients with liver cancer, about 2 years ago, Avastin was combined with an immunotherapy drug called atezolizumab, which is known as Tecentriq. And the combination of Avastin and Tecentriq in patients with primary liver cancer was shown to be very effective in a randomized trial. So we are extremely excited to combine 009 with immunotherapy. We have not done that study yet, but that's definitely something we're planning. And I think ultimately, where the field of oncology is going is exactly what you're asking this question. Combination of angiogenesis inhibition with immunotherapy. If you asked me this question, or any oncologist this question 5 years in the future, I think you'd get a standard answer that that's part of standard therapy in many tumors 5 years from now.
I agree. Okay. What about patients who already have a history of high blood pressure? Would they be excluded from this trial?
So if their high blood pressure is not controlled with blood pressure medicines, then yes, you probably should not go on this study.
Okay. And are you going to be taking patients who have been previously treated with other treatments?
Yes.
They use Topaz and Y90 as examples. That wouldn't matter.
If a patient was in Topaz, they would be eligible for this study.
Okay. Do you think it would be helpful for patients to have or not have certain gene mutations to be eligible for this study?
Sure. So we don't believe that 009 favors one mutation over another. So we believe today that 009 could be used in patients with all types of biliary tract cancers, independent of their mutation status. That's what we believe today. We have not yet proved that, but we believe it could be used in patients with all different types of mutations.
Okay. Will the next phase of the study only be open in the USA, or are you looking into other countries as well?
It's going to begin by opening in the USA. Those are our plans, but we're also evaluating other countries.
Okay. If a patient is controlled hypertension with meds, are they still eligible? You just talked about that.
Yes.
Does the antihypertensive meds affect the efficacy at all?
Not that we know of.
Okay. And a couple others over here. So someone has a fistula in the liver, and the understanding is she would have to be closely monitored if she was in a study with VEGF. Is there a risk with this or any exclusion?
So fistulas in the liver are that's a very, very complicated question, and I think I don't really know the answer to your question, but I think that's a very complicated question. I think probably if you had a fistula in the liver, probably not. You probably would not be eligible for the study.
Okay. I think we answered all the questions. If there's any other questions, please feel free to put them in the Q&A box or in the chat. I have a couple questions for you.
Great.
So would a patient have to fail as a standard of care before they could enter into the trial? Is this a first-line trial? Is second-line trial?
If you don't mind, I'm going to slightly reword your question.
Sure.
If that's okay.
Absolutely.
So when I was seeing patients, I saw almost exclusively patients with leukemias. So the chemotherapy fails the patient.
Right, exactly.
Not the patient, not the other way around. So it's the chemotherapy that fails. This is a second-line study as we've currently designed it. So patients have received frontline therapy with chemotherapy or a combination. So Topaz, the question that was asked about Topaz, that's chemotherapy plus immunotherapy. That would be considered one treatment regimen.
Okay. And this is me with my patient advocate hat on, and I know it throws a kink into your protocol design.
That's okay.
Have you considered a crossover arm for the
So
patients on
we've considered that. At the current time, we do not have a crossover built into the study, because we don't. So why, right? And believe me, I have profound respect for the hat that you are wearing, and I think we don't yet know for sure whether or not 009 works. It's an experimental drug. If we knew that it worked, it would be approved. We have many, many, many hints that it works, and we have hints that maybe it works really well, but we don't know that yet. And it's extremely important that we know that, and the way to know that is in a randomized trial. So we have to know that so that we can figure out whether or not tens of thousands of patients should be given this drug. So I know it's hard. It's so hard because, because
especially
there's,
cancers have not a lot of treatment options,
that's
right?
right. That's right. Plus, every single person is an individual, and you want to give the individual patient the absolute best therapy that you possibly can. And with cholangiocarcinoma, there are so few treatment options, and it's terrible.
Yeah.
It's terrible. And but the job of the FDA is to make sure that these drugs are safe and effective. And the FDA has a really hard job,
Yeah.
They've got a really hard job because, FDA knows that it's all individual people as well, and the folks that we're talking to at the FDA are treating oncologists as well. And they know. So
okay, we had a few
so
more
hard.
questions.
Oh, great. Okay.
We had a few more questions come in. Would chemoembolization affect being eligible for the trial at all?
I don't think so. No, that should not affect eligibility.
Okay. And then somebody asked what a crossover arm is.
Yeah.
I don't know if you want to.
Sure. A crossover arm is when patients in a clinical trial can go from one assigned therapy to another, that they cross over to the other arm. So, crossovers are somewhat controversial, from a clinical study design point of view. Believe me, I get it. The way you ask the question, I for sure get it. But a crossover might not get us the answer.
Yes. Okay. And then could this be used for patients who are currently on targeted therapy like Tibsovo?
Could be.
Okay.
Could be, yes.
And how long is the trial before preliminary results would be known? I guess it depends on accrual.
Wow, yeah, geez. Maybe a year or so, maybe. Maybe, and it depends. It depends how fast patients come into the study. So maybe a year or so.
Okay. When it's ready to be posted on clinicaltrials.gov, I would love to get that information and have you post it on our website so that our patients have access to it.
Sure. That would be awesome. Sure.
And how do we know when phase 3 is open and possible timeframes? So that's what I was going to say. We'll post it on our website. We can let people know as soon as you let us know. And do you have any possible timeframes about when we could expect that?
Probably in the coming months, I hope. Yeah.
Okay.
So it should be in the coming months.
Sounds good. So we will keep you all posted and let you know. Thank you so much for your time today, Dr. Schuetz. It was very, very informative and exciting. We're always looking for more treatment opportunities for sure.
Okay, well, thank you so much. Gosh, the questions were phenomenal. I mean, seriously, the questions were just phenomenal. Thank you so much. This was a great discussion, and I really appreciate talking to all of you today.
Yeah, we appreciate your time and your work in doing research for cholangiocarcinoma patients. And yes, our audience is outstanding. These guys have they always have great questions. I should have forewarned you, but they're always good.
All right,
Thank you
cool.
so much. Everyone have a great day. Thanks for joining us.
Thank you.
Bye-bye.